Hello everyone, this is Bailey Keller with my presentation on the chemistry of forensic blood, serology, and DNA profiling. So, throughout the past 100 years or so, the methods of testing for blood at crime scenes has changed a lot. Today we use presumptive tests, which are basically tests used to see if something is possibly blood, and confirmatory tests, which are used to confirm if the blood is actually blood or not. Confirmatory tests have to be done in order for blood to be used as evidence in court cases, which confirmed blood can then also be used with PCR to make DNA profiles and then uploaded to databases which are used across the country for DNA evidence. So essentially the development of these tests is called blood serology, which is the study of the specific detection of the contents of blood, such as hemoglobin and antibodies, where serologists develop the test so investigators can detect the presence of hemoglobin and such. So first we'll talk about presumptive tests, which are used to detect the possible presence of blood at crime scenes so they can be collected and sent to labs for analysis. Two of the more common ones are the luminol and Kastle-Meyer test. You may have seen the luminol test on like CSI and stuff, where it's essentially just a blue glowing color, which we'll talk about in the next slide. And also two other tests I forgot to mention are the leukomalachite green test and the HemoSticks, which are also tests, but they're not used as commonly as the other two I mentioned. Essentially, all these presumptive tests use this very similar mechanism, a peroxidase reaction, where they all have kind of the same chance for false positives due to their sensitivity. Surfaces with bleach, metals, oxidizers, or even where vegetables are stored can lead to positive results due to how the test works, where they kind of just need an oxidizing agent to trigger the reaction. And also these presumptive tests will not tell the difference between animal blood and human blood, which is why the confirmatory tests are used especially. So as I mentioned, luminol, which is a sprayed chemical that exhibits chemiluminescence when in the presence of an oxidizing agent. So when a surface covered in blood is sprayed with luminol, the iron in the hemoglobin acts as a catalyst, where the iron molecule will cleave one of the oxygen molecules in hydrogen peroxide. Then the hydrogen peroxide molecule will go to the luminol molecule, as you can see on the right, and it'll remove those two NH groups and replace them with oxygens, and that makes it charged and causes it to exhibit light through, as I mentioned, chemiluminescence. The luminol is a very fast test and very efficient, detecting 10 to the negative 6th molar to 10 to the negative 8th molar solutions of hemoglobin. Although there is a con with luminol in that too much of it being sprayed on a sample can dilute the blood and make it unusable for DNA analysis. The next is the Kastle-Meyer test, which is done by getting your sample of suspected blood onto a piece of filter paper and swabbing it with a wet swab. Now 2 drops of reagent are added, which is essentially just a reduced phenolphthalein reagent, which is colorless, and then if it has actual hemoglobin on it, which will serve again as a catalyst. It reacts with hydrogen peroxide and the reduced phenolphthalein. It'll oxidize it into the pink colored phenolphthalein that we've seen before. And so obviously when it turns pink, that'll mean it's positive for the possible presence of blood. This is a very sensitive test in that it'll detect percent concentrations of blood at 0.1%, 0.01% actually. Although, as I mentioned, it does have the potential for false positives with other oxidizing agents. So now the leukomalachite green test, which uses a reflex reagent of malachite green. Then H2O2 is added to the sample with reagent. If the color changes to a deep green-blue color, similar to the one on the left, then it is positive for the possible presence of blood. And if there's no color change, then it is negative. This is also very similar to the Kastle-Meyer test, although It has a sensitivity of half what is expected from the Kastle-Meyer test, although it is still used in some circumstances. Finally is the Hemastix test, which uses a reagent of 3,3,5,5-tetramethylbenzidine. This reacts in the same peroxidase method mentioned from the other tests, although it uses different colors to show the concentrations of hemoglobin. So the greener color of the sample represents a higher concentration, which is handy if you need that, and with a sensitivity of 0.015 to 0.062 milligrams per deciliter for hemoglobin. So it's very sensitive. Although this test tends to be more expensive than especially the luminol test. So now we get to the confirmatory tests, which these are important as the presence of blood needs to be completely verified so it can be used as evidence. These are not used in place of presumptive testing as they are much more expensive and take a lot longer than presumptive tests, although they will conclusively identify the substance with a much smaller risk of false positives. So we'll talk about two here, the ABA card hemotrace and the Takayama crystal assay. So first, the ABA card hemotrace is a confirmatory test that tests for the presence of human hemoglobin. So obviously this is beneficial as it is human, and it can tell the difference between animal hemoglobin. So the test starts with your blood sample being added to the S well in the bottom there, where antibodies will react and form a complex that will remove to the T well, where there is more antibodies and also a pink dye. If the pink line forms in the T zone and the C area, which the C area serves as your positive control, then the test is positive. Although if it's only the C area, then it's negative, and if there's obviously none from the right side one, it's an inconclusive test, so it'll be probably performed again. This test is very accurate, although there is a chance for false negatives, which occurs with excess hemoglobin, where the concentration of hemoglobin is too high. It prevents the complexes mentioned from forming with the antibodies, so no pink lines are formed essentially. And also there is a chance of false positives in the presence of ferret blood, interestingly enough. Apparently ferret blood is very similar to human blood. Didn't know that. So next is the Takayama crystal assay, which is a chromatographic test wherein the presence of blood and with pyridine and glucose reacts to the hemoglobin. Essentially what happens is the pyridine reacts with the hemoglobin molecule and causes the formation of pyridine ferroprotoporphyrin and a hemochromogen product. Then glucose causes the hemochromogen product to precipitate out of solution in the form of the crystals that you can see here in the picture, which are pretty cool looking. The formation of these crystals takes a while to perform, and if the temperature of the solution decreases, then they will go back into solution. So the temperature has to be constantly controlled for multiple hours. If the crystals are visualized under the microscope, then the test is positive. Easy enough. So now we finally get to DNA profiling, where after the blood is confirmed, the DNA is extracted in a process called PCR. So this is a 4-step process of extraction, quantitation, amplification, and electrophoresis. So first the DNA is extracted from the blood, and now quantitation is performed by a computer to count the amount of DNA that was extracted. Essentially, this is done to make sure you have enough DNA to actually perform PCR. Only a nanogram of DNA is actually needed to start the process. So now the DNA is heated, which causes the structure of DNA to denature and lose its quaternary structure, making it a single-stranded DNA. Then primers are added to the solution, and this starts the DNA replication process, which causes it to replicate into multiple strands and can be repeated 20 to 30 times to produce millions of copies of DNA in only a few hours by a machine called a thermocycler. So now the DNA is separated using gel electrophoresis, or it's separated by size. Most of the DNA is made of long stretches of nucleotide pairs with these short repetitive base pair sequences called short tandem repeats. Essentially, these repeats are inherited and easily detected in in the DNA, and they're very useful for identifying markers within a person's genome. So essentially, the machine, as I mentioned earlier, quantifies these STR markers, short DNA repeat markers, and can make a profile, DNA profile, as you can see on the photo on the right. So then these profiles can be saved into uploaded databases, which I'll talk about in the next part. So these DNA profiles are unique to every single person, with a possible 1 in 10 billion chance of a false positive result. So as you can see, the suspect 2 matches up perfectly with the evidence sample on the right. So that person brought in for questioning. So now we get to the CODIS and NDIS, which is essentially the database that all these DNA profiles are stored in. So the way it works is that each individual lab will upload them to their local database of DNA profiles, which is called just basically the local DNA index system is what LDIS stands for. Then they will upload this to the state level, and then eventually the national level, so that way these DNA profiles can be seen by police departments across the world, not across the world, across the US. So in conclusion, this is a very evolving field where different tests are being tested every year to see which are more effective than the other. As of September of 2020, nearly 20 million of the DNA profiles have been stored in NDIS, which have assisted in more than 500,000 investigations across the United States. A recent example of this is back in 2018. The use of the NDIS caught the Golden State Killer, who killed 13 people in California more than 40 years ago. And as I mentioned, he was finally arrested in 2018. So things like this can be, these cold cases that were closed years ago can now be closed thanks to the benefit of the NDIS. So the future of blood serology and advancements in presumptive and confirmatory testing will lead to even more DNA profiles in the NDIS, which can be used to solve cold cases for years to come. Thank you.